Hi, my name is Callie Weber, and this is my project on how decreased male blood-brain barrier permeability lowers aqueous glucose uptake. So Alzheimer's disease is associated with a global decrease in glucose metabolism and increased blood-brain barrier permeability. So what we see here is the fluorescent glucose uptake, and this is shown in a PET scan. We have in the top row a healthy individual, in the middle row someone with mild cognitive impairment, and in the bottom row someone with diagnosed Alzheimer's disease. And what we see marked by a decrease in the amount of red or warmer colors is a decrease in glucose uptake with the diagnosis of Alzheimer's disease. Additionally, associated with Alzheimer's disease is the leakiness of the blood-brain barrier. So what we see here is a healthy capillary that is surrounded by endothelial cells in blue forming the blood-brain barrier, pericytes in green, and then astrocyte feet in orange. And you can see that the solutes and the red blood cells are contained within the capillary. In Alzheimer's disease, however, there is disruption of this blood-brain barrier, and this leads to the leakage of the solutes into the brain from the capillary channel. And associated with this leakage is a decrease in the pressure drop across the capillary wall. So generally there's a higher pressure in the capillary and a lower pressure in the interstitium. However, if the blood-brain barrier is disturbed, this leads to a decrease in the capillary pressure, and this leads to a decreased pressure drop across the capillary wall, and this will be discussed further in the project. When you look at the brain endothelial cells that form the blood-brain barrier, we have some data published by Holman et al. In 2017, and this shows that the resistance across the blood-brain barrier is greater in females than it is in males, and this is indicative of greater integrity of the blood-brain barrier in females than in males. And this is just in baseline healthy cells. So this project is based off of a paper published in 2011, and it's titled The Application of the Kedem-Kachalski Equations to Membrane Transport of Ethyl Alcohol and Glucose. And this paper examines the equation that you see here, which is the Kedem-Kachalski equation, and it specifically studies how the components of the equation impact the volume flow across a membrane. And this volume flow is this JV that is in the equation you see here. In this paper, they use this model of feed, a permeate, and a membrane. So you have the feed, which is the source of the ethyl alcohol or glucose in this paper, and then the permeate, which is where it is traveling to, and a membrane separating the two. And then we have the hydrostatic pressures of the feed and of the permeate, marked by P1 and P2, and we have the concentration of the feed and the permeate marked by C1 and C2. Adjusting this to work for what would be happening in the blood-brain barrier situation that I will be demonstrating in this project, our feed is going to be our capillary, our membrane is going to be our blood-brain barrier, and the permeate is going to be the brain. So the aim for this is to model differential volume flow of aqueous glucose based on sex. So here we have the diagram of how this will be modeled. What's important here is that we want to model this differential volume flow of aqueous glucose based on sex so that we can determine how the integrity of the blood-brain barrier specifically impacts glucose flux and how this could be related to the development of Alzheimer's disease. So here we have the Kedem-Kachalski equation for volume flow written out along with what all of its components are. So the equation is the volume flux, which is JV, and that is equal to the filtration coefficient of water, which is the amount of water filtered across the membrane per unit time, and that's multiplied by the kinematic viscosity of water times the density of water divided by the kinematic viscosity of the aqueous glucose solution times its density, and this is multiplied by the hydrostatic pressure drop across the membrane minus the reflection coefficient times the osmotic pressure drop across the membrane. And so the factors that will be affected by the permeability of the membrane specifically are the filtration coefficient of water. So the leakier the membrane is going to be, the more water that's going to be filtered across it per unit time. And then the hydrostatic pressure drop across the membrane. So like we were discussing with the disruption of the blood-brain barrier in Alzheimer's disease, a weaker blood-brain barrier is going to have a lower hydrostatic pressure drop across the membrane. So in order to apply this model to our blood-brain barrier, we are going to have to make some assumptions and simplifications that make this model valid. So the first two assumptions are ones that are necessary for all Kedem-Kachalski models, and that is that both sides of the membrane are sufficiently dilute and well stirred, and that the solutions are isothermal. So for this, all of our solutions we're going to be assuming are at 37 degrees Celsius. And then in order to make our blood-brain barrier model fit with this equation, we are assuming that the blood-brain barrier can be modeled as a homogeneous polymer membrane. In reality, this would be a little bit different because glucose would have to cross both the capillary and interstitial sides of the endothelial cell membranes, but we're just going to be modeling it as one homogeneous polymer membrane. Um, the second and then that blood-brain barrier endothelial cells use a negligible amount of glucose for their own metabolism, so the blood-brain barrier cells would use some of the glucose that they take up from the capillary in their own metabolism. However, we are going to assume that this is a negligible amount and that all of the glucose taken up by these capillaries is then shuttled across the barrier into the interstitium. And then finally, we are assuming that the system is diffusion limited, so this would mean that the number of glucose transporters in the cell membrane is much larger than the number of glucose molecules that are diffusing. And this is just because if the number of glucose transporters were less, that would make the glucose transporters the rate limiting step in the equation rather than the glucose diffusion. So looking at this equation, we have the water filtration coefficient, and that is this LPW coefficient. And this is dependent on the permeability of the blood-brain barrier and therefore is sex-specific. So this LPW, Paulson et al. Has found that it is equal to the filtration permeability coefficient times the molecular weight of water times the capillary surface area per unit of brain, and this is divided by the ideal gas coefficient times the temperature. And so this paper by Paulson et al. Found the filtration permeability coefficient to range from about 9.4 to 11.4 times 10 to the negative fourth centimeters per second. So then this leads to the LPW calculation, which is then calculated to range from 6.56 times 10 to the negative seventh to 7.97 times 10 to the negative seventh. And for the sake of our project, we are going to assume that the lower water filtration coefficient corresponds with the female case, and the higher filtration coefficient corresponds to the male case. And this is because we are assuming that the male blood-brain barrier is leakier, and this would increase the amount of water that's being filtered across the barrier per unit time. And we are assigning the male and female values to these high and low found in this range from this Paulson et al. Paper because there is not data on this that has been calculated for how these filtration permeability coefficients would vary with sex. So the first portion of this experiment was to look at how solute filtration is affected by increasing the solute concentration. And so this is based on this equation that you see here at the top, where we have that the membrane permeability to the solute is equal to the membrane permeability of water times the dynamic viscosity of water divided by the dynamic viscosity of the glucose solution. And in order to calculate this, we had to look into calculating the dynamic viscosity of glucose, which I used data from a paper of Weatherby et al. And this shows how the dynamic viscosity changes with glucose concentration. So that's what you see in this graph here, where you have an increase in dynamic viscosity with increase in glucose concentration. And then to calculate the dynamic viscosity for every glucose concentration, I fit a quadratic to this data set and then used that to calculate the dynamic viscosity for each concentration. I then inserted that into this LP equation in order to calculate the LP for every concentration. And then I plotted LP as a function of concentration. So what you see here on the x-axis, we have the glucose concentration, and on the y-axis is the filtration coefficient of the solution, so that's the LP. And you can see that my data set pretty closely matches what they published in the paper for the ethyl-alcohol relationship with concentration, which is what you see here on the top right graph. But basically what this means is that as the concentration increases, the membrane permeability to the solute decreases, and this makes sense because the membrane permeability is taken with respect to concentration. So if you're increasing the concentration but keeping your flux across the membrane the same, you're going to have a decreased permeability constant. And this LP is higher in all male simulations than female simulations, and this is just a factor of the tighter barrier strength in the females, showing that you would have a decreased filtration coefficient for every glucose concentration. So this next portion of the project looks into how volume flow is affected by a changing filtration coefficient. So what we have here is LP as a function of volume flow, and this is looking with no change in hydrostatic pressure. So if you remember the Kedem-Kachowski equation, you have the change in hydrostatic pressure minus the change in osmotic pressure. And so this is just based on the change in osmotic pressure. And you can see that, similar to the results that are published in the paper here on the right, our results show that there is an increase in volume flow with a decreased filtration coefficient of the solution. And you can see that the males are much higher than the females, and this means that for the same volume flow of the solution of the glucose across the membrane, there has to be a much higher filtration of water across the membrane. And then we repeated these results, but looking at LP as a function of volume flow with no change in osmotic pressure. And so for this part, we got rid of the osmotic pressure. So this is just the change due to the hydrostatic pressure. And you see very similar results. However, of note here is that the scale of these two graphs, if you're just using osmotic pressure, you have a scale of 10 to the negative ninth. However, if you're just using the hydrostatic pressure, you have a scale of 10 to the negative fifth. And so this is indicative that the osmotic pressure has a much lower influence on the relationship between LP and volume flow than the hydrostatic pressure does. So basically what these graphs are showing is that females have more efficient filtration of glucose across the membrane than the males. So then finally, I combined those last two simulations, and this is LP as a function of volume flow, and this is using both changes in hydrostatic and osmotic pressure. And this graph actually ends up looking exactly the same as the graph for the LP versus volume flow using only hydrostatic pressure, and that's because the osmotic pressure change was so small that it was not a driving force in this scenario. And that makes sense because the change in concentrations that are driving this osmotic pressure in the brain are really from about, in a capillary, you would expect to have about 5.5 millimoles of glucose per liter. And then in the capillary, you would expect to have, or in the interstitium, you would expect to have about 1 millimole per liter of glucose. And this change is just too small to really drive a huge flux of glucose across the membrane. So again, just reiterating that this is showing that the females have more efficient glucose flux across the membrane. The final portion of this project is on how shear stress affects the efficacy of filtration across the blood-brain barrier. And so shear stress is often used as a proxy for exercise. There's preliminary research that shows that exercise can actually slow the progression of Alzheimer's disease. And one of the thoughts is that by exercising, you're increasing your blood flow through your brain and increasing the transport of glucose across the blood-brain barrier for the cells to use as metabolism, for their metabolism. And so in this, we're looking at how shear stress can impact the flux of glucose across the blood-brain barrier. And in order to do this, increasing shear stress is like increasing the force. And so this increases the capillary pressure because you're having more force driving your system, so you're having a higher capillary pressure. So the average hydrostatic pressure in a capillary is about 2,200 pascals. So for this experiment, I set the delta P in the low shear stress case to be 0 to 3,000 pascals, encompassing this average. And then I doubled that for the high shear stress case to look from 3,000 to 6,000 pascals as the pressure change across the blood-brain barrier. And I used these values because there is not necessarily a numerical relationship that has been derived between the capillary shear stress and its resulting hydrostatic pressures. So hopefully future research could provide numbers that will elucidate this numerical relationship and allow us to further study the effects of shear stress on glucose transport. However, for our simulation, we just use this 0 to 3,000, 3,000 to 6,000 set. And what you see is that with increased shear stress, you have increased glucose flow across the membrane without necessarily changing the filtration coefficient. And this is similar for males and for females. You see the males still have a much higher required filtration coefficient, so you're having decreased efficacy of glucose filtration across the membrane. However, for both situations, you're seeing increase in volume, you're seeing a higher volume flow with the same filtration coefficient, indicating that you are having increased glucose transport across the membrane. So in conclusion, this study has shown that males have a generally higher filtration coefficient of aqueous glucose across the blood-brain barrier than females. And what this means is that males have a higher it takes a longer amount of time for males to have the same glucose filtration across the membrane. And so this indicates a decreased efficacy of volume flow of aqueous glucose across the blood-brain barrier. Increasing the shear stress is one mechanism by which filtration across the blood-brain barrier can be increased, and this then increases the glucose availability to the brain, which can mitigate the symptoms and the progression of Alzheimer's disease. So this is a computational model, and it's based on a lot of experimental data that's extrapolated from papers that are not exactly measuring transport across the blood-brain barrier. A lot of them are based on like pressures in capillaries in the body and that are not blood-brain barrier specific. So further research should be done to discover the exact values for the reflection coefficient for glucose across the blood-brain barrier, water filtration coefficients in males versus females. If you'll remember, the data I used was just a high and low water filtration coefficient in general, but it would be nice to have the actual values for males versus females. Hydrostatic and osmotic pressure differences between brain capillaries and the surrounding tissue, and a numerical relationship between brain capillary shear stress and its resulting hydrostatic pressures. And future computational studies could be improved by modeling the blood-brain barrier as a two-membrane system. We did a simplification to just model it as one membrane. Taking into account the relative abundance of GLUT1 transporters in males versus females, there is some preliminary data that show that females might have increased GLUT1 expression, and so so you could do adjustments based on that. And then accounting for the percent of glucose that's used in endothelial cell metabolism versus what is passed on to the rest of the brain. And that's my presentation. Thank you.